very much for this opportunity. And we are actually in a clinical trial with our human neural stem cells in a rare lysosomal storage disorder called neuronal ceroid lipofuscinosis, or Batten's, is the more common name. So I'm going to actually take a different tactic and actually go through what it took to get to the clinic. Wasn't easy. And just to make sure that everybody knows, the reason we are working with these tissue-derived adult stem cells is because when we started this work, the human embryonic stem cell had not yet been identified. So this goes back a long way before either the embryonic stem cell or the iPS cells were around. So we have published on the identification and banking of human neural stem cells. And it's a two-step process. You first identify and purify the cells by using surface markers and a high-speed cell sorter. This process purifies the cells from brain tissue. The cells are then grown in a serum-free defined media, and they grow as balls of cells known as neurospheres. And using this two-step process allows you to get a very reproducible expansion of the cells and growth so that you can bank the cells. And that's going to be where it's important to get your biological data, but when you start to manufacture these cells for clinical use, it becomes very important that you can do this reproducibly. Growing in these conditions, these cells maintain normal karyotype. They don't form tumors in vivo, and we've transplanted over 3,000 of these. They retain their multilineage potential, which I'll show you. And they don't require pre-differentiation before you transplant the cells. So we can assess the purity of the cells prior to transplantation. And those of you who are marching toward the clinic, you'll know that that purity issue is a big question the FDA will ask, the regulatory bodies will ask you. We've shown that at the single cell level, these cells self-renew. So if you start with a single cell in a well, these will form these balls of these neurospheres. And then if you force them to differentiate, they'll form neurons and glial cells. So that's the reason we've elevated to the stem cell status, because we've shown at the single cell level, they expand and maintain their multipotent potential. In animal models, these cells engraft robustly and migrate throughout the brain, and it doesn't show up very well here, but all of that brown staining you see are the human cells that have engrafted, migrated throughout the brain after a single transplant. The cells take up residence in the endogenous mouse stem cell niche in the brain, and they proliferate very slowly, so it's restricted proliferation. They migrate along the normal pathway that the mouse neural stem cells take, called the rostral migratory stream. And there they land in the olfactory the olfactory system, where they mature into granular neurons. So in a very site-specific manner, these cells mature. And on this slide, I'm just showing you that depending on what part of the brain you look at, you find neurons, astrocytes, and the myelin-producing oligodendrocytes. And I won't go through all the data, but we have shown that this is a very reproducible engraftment and site-specific differentiation. So we've taken this process and we've adapted it to a GMP cell banking production, where we start with qualified raw materials, which is very important, that you have the highest quality of materials, hopefully that are not animal-derived or containing any source of potential viral contaminants or anything like that. The process is done in a GMP cell processing facility under containment asepsis, using all of the controls that you need in a GMP cell processing. And I think it's key to know that, if you develop your cells, you know the best how to grow them. And so we were very lucky to identify a GMP manufacturing site where we actually grow the cells there. It's our staff that do that. Because growing fibroblasts is one thing. Growing your cells is quite a unique skill. We've also developed the assays for characterization, because when you get those cells out of your cultures at the end of the however many day processes it are, and then you're making the patient product, you have to be able to characterize it and show it it's the same this time and every time that you've made the cells. We've also done all of the release testing that the agencies, the regulatory bodies require, and that's required on the master banks and the working banks. And this release testing takes 4 to 6 months. You also have to do rapid release testing so that when you're ready to manufacture the patient product, that you can show your cells are sterile, endotoxin-free, et cetera, purities. So all of those assays have to be developed and demonstrating the characterization of your cells. Another important point is the lot-to-lot comparability, because unless you have an endless supply of the cells, which you might in the case of the embryonic stem cell, you'll have to go back and make a new master and and so you have to demonstrate so that you don't start all over again with all the safety testing in a phase 1 trial, that the lot you made 5 years ago is comparable to the lot you're going to make 5 years from now. So it is a very, very long road, many pitfalls in between. So I've just talked briefly about the attributes of these human neural stem cells. They make the 3 cell types of the CNS. And so when we started to think about what avenue we pursue for a clinical trial, there were many things. In vogue at that period of time was Parkinson's, but there were a lot of people who were able to cure Parkinsonian animals, but humans, the data was iffy. So we thought, okay, we could use these cells to neuroprotect through the delivery of soluble proteins, such as lysosomal enzymes for one, or neurotrophic factors. We can neuroprotect through remyelination of host cell or host neurons. And we can have neuronal replacement, such as in Parkinson's. And so after many debates and meetings with experts, we decided to pursue the pathway of looking at the cells in the lysosomal storage disease that affects the central nervous system. It was called the lowest hanging fruit because it didn't require neural circuitry integration, right, making the proper functional connections. The transplant of the cells directly into the brain or into the CNS circumvents the blood-brain barrier. So you could address these lysosomal storage diseases that affect the central nervous system, where enzyme replacement therapy wasn't effective. Our hypothesis is that these normal human cells, these donor cells, could serve as cellular mini pumps to deliver the enzyme throughout the CNS. It offered the prospect of a one-time intervention where you're delivering these cells, and because these are stem cells and they're able to divide and make all of the cell types, that it could provide a continuous reservoir of the enzyme. And it also offered the prospect, if you really did make new neural cells, that you could have neural replacement and repair the damaged brain. Most importantly, there were good animal models that recapitulated the human disease that allowed you to demonstrate proof of principle for that particular disease target. So here's a cartoon of what we thought would happen for these lysosomal storage disease. So in the Batten's disease, what happens is you have a deficiency of a normal housekeeping enzyme, and the lack of that enzyme causes the buildup of storage material, and this lipofuscin storage material accumulates in the brain and causes the neuronal cell death. These are housekeeping enzymes, and so cells produce these proteins, and 10 to 20% of these lysosomal enzymes are just naturally secreted outside the cell. So our hypothesis is that these human neural stem cells would secrete these lysosomal enzymes. The lysosomal enzyme could be taken up by the deficient cells, and that through that process it would reduce the storage material and neuroprotect those host cells. So we had shown in a transwell system similar to the one that Dr. Hint just described, that our neural stem cells, when co-cultured with patient fibroblasts, would produce the enzyme, and that could be uptaken by these deficient human cells. So that was the in vitro proof of principle of our cells. What happens when we put these cells into the animal model? So, as I mentioned, we decided on the neuronal ceroid lipofuscinosis. They're a progressive neurodegenerative disease of children. So the children are born, they develop normally until 6 to 18 months if you have the infantile form, or 2 to 5 years if they have the late infantile form. And because they have genetic mutation of these lysosomal enzymes, the autofluorescent material starts accumulating, and their neurons start to die. We were particularly interested in two forms, the infantile and the late infantile form, because there were known genetic mutations. There was an enzyme assay for the deficient enzyme, and there were good animal models that recapitulated the human disease pathology. And it is a rare disease. The incidence is 1 in about 12,500 births. So it was a rare and fatal disease. And just to give you an idea of the devastation of the disease, on the left, on your right, you see the brain of an NCL patient. And what you'll notice is that the ventricles are significantly enlarged, and that's due to the atrophy of the brain. What happens is you just start filling up with the CSF fluid. So compared to the normal brain, you see that the ventricles are very enlarged, and you see more pronounced sulci and gyri, because the brain is just degenerating. So in the animal model of the infantile form of the disease, if you look at the hippocampal area, it's a nice area to do some quantitative analysis of the number of neurons. On your left, you see the same area of a normal mouse, the non-skin mouse brain. And you can actually count the number of neurons in this particular section of the brain. So when you count the number of neurons in this part of the brain and you compare it to the knockout mouse, which lacks the enzyme for the infantile form, you see that only 8% of the neurons that should be present in this area remain at this point in time. So it's significant neuronal loss. When we transplant the human neural stem cells into the brain of these animals, we're able to neuroprotect the host cells, and those are shown here in this graph. So a low dose of cells will protect about 30%, high dose of cells about 60%. And if you look at the corresponding histology of the brain, you can see that there is significant protection of those neurons in the hippocampal CA1 region. So these cells, when delivered, when transplanted directly into the brain, can neuroprotect the host cells. So when we started looking at the ability to move forward, we had our pre-IND meeting with the FDA, and of course they have requirements of what you need to demonstrate. Their primary concern, again, is safety. Human neural stem cells have never been transplanted into the brain of patients. Fetal tissue has, but not purified, expanded human neural stem cells. So we initially filed our IND, and as we have publicly announced, we were put on hold because we had to demonstrate more things to convince them of the safety of these cells. Suffice it to say, we managed to convince them that the cells were safe. We provided them with all of the banking data, the proof of principle of these cells in the animal model for Batten, which included the engraftment, the migration, the enzyme production, the neuroprotection of host cells. And we did extensive safety testing of these cells, both in murine and primate animals. And one of the problems moving forward with cell transplantation, in animal models, you're going to be doing a xenotransplant. So immunosuppression isn't effective for the xenotransplant component. So it's hard to immunosuppress the animals to the level you need to get across xeno barriers. But in humans, you'll be doing allotransplants, which are not as tough as a xenotransplant barrier. So what is going to be the appropriate animal model for cell therapies moving forward? We use the immunodeficient non-skin mouse. If they want more than one species, what is your second species? I think that's a question that everybody is going to struggle with. Somebody will make lots of money if they define another larger animal that is immunodeficient. And we've been looking. Arabian horses are immunodeficient. So, right. Yes, well, that's what, and it's not widely available. You can't do the number of transplants that you need to do to demonstrate safety. So we have initiated a phase 1 trial in patients with either the infantile or the late infantile form of NCL. It was an open label study with a low and high dose cohort. The cells were directly transplanted into the brain, either cortical injections or intraventricular injections. The patients received 12 months of immunosuppression. Of course, the primary endpoint was safety. Secondary endpoints are measures of efficacy. We did have an independent data monitoring committee so that they assessed the safety of the transplants prior to transplanting another patient. The current status is that the first patient was transplanted in November of 2006. The last subject was transplanted in January of this year. 3 patients have completed a 1-year evaluation. 1 subject died due to underlying disease. This was also again assessed by the independent data monitoring committee and the FDA. So we expect that after a 1-year follow-up, the data will be locked in January of next year. All of the patients who completed a 1-year evaluation roll into a long-term safety study, which monitors the patients for an additional 4 years. This trial, this long-term safety study was initiated in November of last year, and so far 3 subjects have enrolled. So what's on the horizon? So I talked about the neuroprotection through the delivery of soluble proteins. We know because these lysosomal enzyme or housekeeping enzymes, you would expect them to produce all of the other lysosomal enzymes. We've tested 3 to date and shown that these cells produce the enzymes for Sanfilippo, Hurlers, and Gaucher's. We've also been exploring the use of these cells in retinal degeneration disorders, and we have a collaboration with Peter Francis and Ray Lund at OHSU in Oregon. We also have extensive data on the neuroprotection of host cells through remyelination. We have a collaboration in spinal cord injury with Eileen Anderson and Brian Cummings at UC Irvine, and they've been able to show that animals who received spinal cord contusion injury are able to have a locomotor improvement, and that improvement is directly linked to the presence of these human neural stem cells, which have differentiated into oligodendrocytes and remyelinate host axons. They've also differentiated into synapse-forming neurons. So that work is proceeding forward. We have extensive data in a mouse model, a hypomyelinating mouse model called Shiverer, and one of the disease targets we're considering is this rare fatal Pelizaeus-Merzbacher, in which these patients have a dysmyelinating disease. So thank you, and I'll take it.
That was wonderful.
What can you tell us about how the 5 patients are doing?
So we're doing an interim data analysis on the first cohort, but the data isn't locked yet for that interim. So as soon as we have something, we will be discussing it. To date, there are no safety concerns either by the images that we looked at or there's nothing that couldn't be attributed to the disease course.
What's the normal life expectancy?
So a lot of it depends on the care that the patients receive. They live into their late teens if they have the late infantile, early to late teens. Yes.
Now they've launched clinical trials. Do you have a sense of how much time and cost it's going to take for the lysosomal storage specifically?
How much it's going to cost for this particular disease, the Battens?
Yeah, or other. Are you going after other lysosomal storage disorders as well, or just that was Batten?
Well, so that is a good question because the NCL is rare. The most prevalent is the infantile form in Finland because it has a 10-fold higher rate in Finland than it it does in the US. It sort of depends on what kind of feedback we get from the regulatory agencies as to how much preclinical animal data we have to do before we enter into another disease target. Because if we know that the enzyme is produced for San Felipe, we have to go back to the animal model if you have human safety data. So,
Do you have a sense compared to other, I mean, a company like Genzyme, which has gone, done a lot of work with LSD, is there any comparable, to their clinical trial timelines and costs?
well, so there's a difference, remember, because this is a single transplant of cells, whereas there's repeated weekly dosing, okay? So you manufacture the cells and you do the transplants one time, hopefully. And so it's a very different paradigm. And you can't really translate the enzyme replacement therapy to the cell therapy. Certainly in terms of numbers of patients, if you go back and look at all the lysosomal storage diseases and the number of patients it's taken to get approval by Genzyme or BioMarin, you're not talking about hundreds and thousands, you're talking about less than 100 in most cases.